Hello, and thanks for joining us. This is the first segment, the first of 4 segments. So this is a large chunk of lecture material. This is the first of 4 segments covering cells and movement across the plasma membrane, and we'll spend several weeks on this lecture topic. This chunk of material covers slide number 1 through number 43. This is an overview of all of the topics in the entire lecture, all 4 segments. And in this first segment, we will cover the first 2 shown here. I will give you an overview of cell structure and function, and we'll talk about some of the details of protein synthesis. That is, how proteins are created inside of our cells. Today is a beautiful day to learn physiology, and I'm glad you're here. One way to think about you as an organism is that you are a collection of cells, roughly 30 trillion cells, and they're all working together to make up what we see and what you think of yourself. Your entire body is made up of cells and materials that are secreted by cells. And secrete is a word that we'll use often this semester. When a cell secretes something, it means that it manufactures the substance and then it expels it from the cell into the extracellular environment. So here's where I ask the class if there are parts of your body that are not composed of cells or materials secreted by cells. Is there anything about what you consider to be your body that doesn't conform to this statement? And some people think about it, and eventually a hand or 2 go up. And then I ask the class, well, do you have any fillings in your teeth? Does anybody have any metal plates holding bones in place? Does anybody have any tattoos, for example? And then many hands go up. I myself, I have a tattoo. I have fillings in my teeth. I have a plate that's holding my radius together. So lots of us have technology added to our body. But there are usually a few students in class that say no to all of those questions. And so this statement holds true for some of us, but not all of us because of technology. So this is an image of a generalized animal cell. It's not intended to represent any one type of cell in your body, but rather it's intended to represent all of them because all of our cells are eukaryotic cells. They have nuclei and they have organelles, these little compartments inside of them that are dedicated to specialized structures. They all have a cytoskeleton. And so we'll use this generalized animal cell to start to understand the basic structure of all of our cells. And then down the road, we will begin to talk about very specialized cells, things like muscle cells and blood cells and neurons. So most basically, a cell is a container, and the cell is surrounded by a membrane known as the plasma membrane. The plasma membrane is the exterior coating around the cell, the wall of the container, so to speak. And then inside that container, we find a liquidy material called cytosol. It is a body fluid, an intracellular fluid. And then suspended in that cytosol are organelles. These are membrane-bound containers that are dedicated to specialized function. They're like mini organs. That's the analogy here. So you have a urinary bladder, you have a gallbladder, you have a stomach, you have a heart. Those are organs inside of you, and they are all containers. They're hollow. We have those same hollow containers inside of our cells where specialized activities occur. And we'll talk about some of them. And then we also have structures within the cell that are not containers, things like ribosomes and the cytoskeleton. So how big are human cells? And what tools can we use to visualize cells and subcellular structures? So we can't see human cells using our naked eye because they're just too small. Even the largest human cell is right at the human egg cell is right at the boundary of what the human eye is able to see. So we use microscopes to look at cells, and perhaps in your high school biology class or maybe in some of your college biology classes, you used a light microscope to look at cells and tissues. And I love histology. So throughout my lectures, I do pepper them with histology images, photographs taken using a compound light microscope. And then there are electron microscopes. There are a couple of different types of electron microscopes. I myself have never used an electron microscope. It's a sophisticated piece of machinery, and I've never been fortunate enough to work in an institution where I was invited to use the electron microscope. In my research I've never had a need for an electron microscope, but the images that they produce are fascinating. And I will also pepper lectures using images taken from both transmission and scanning electron microscopes. And then we can even see down to the level of molecules, and we can visualize individual atoms. As technology improves over the years, we now are able to use microscopy technology to see individual atoms. It's pretty cool. So what are the structures that we're looking at here as we go smaller and smaller? So we'll start with that typical human cell. It's not meant to represent the largest cell. It doesn't represent the smallest cell, but rather it represents an average human cell. And then the next three structures are all structures that occur inside of a human cell. The mitochondrion and the lysosome, those are organelles. They are surrounded by a membrane. They are containers. And very specific things happen inside those two organelles. Ribosomes are not containers. It's not a hollow structure. Ribosomes are made out of a type of RNA. And we'll talk more about ribosomes when we talk about protein synthesis. So all three of those structures occur inside of our cells, and then moving toward the right, we see the plasma membrane. And the plasma membrane is the membrane that specifically surrounds the exterior of the cell. And the plasma membrane is made out of molecules. And what we can see right there is a double layer of phospholipids. And we'll talk extensively about the structure of the plasma membrane in a little bit. And then moving toward the right, we see increasingly smaller structures. We get to a protein. Proteins are molecules. I hope you remember that they're made up of amino acids, and proteins are everywhere in our cells. They're in the plasma membrane. They're in lysosomes. They're in mitochondria. They make up the cytoskeleton of the cell. It's very difficult to get away from proteins if you wanted to in a living system. And then, of course, all the way to the right, we see the smallest thing on this scale, the smallest thing really that we'll pretty much ever think about in physiology, although we do think about subatomic particles, right? Protons, neutrons, and electrons. The smallest atom is a hydrogen atom, right? Because it has an atomic number of 1. There's just one proton in the nucleus of a hydrogen atom, and it is one of the 4 most abundant chemical elements in the body, and there's lots of hydrogen in that protein molecule, for example. There's lots of hydrogen in each one of those phospholipid molecules that make up the plasma membrane. So here's our first microscopy image. This is a photomicrograph that was taken using a transmission electron microscope. And what we see here is a fraction, a piece of one cell, where we're looking at part of the contents of that cell. And so up top, you can see the nucleus along with the membrane that surrounds the nucleus, sometimes called the nuclear membrane or the nuclear envelope. And then below that, outside the nucleus, we see what's known as the cytoplasm. That's the cytosol, the liquidy fluid inside a cell with all of the organelles and other structures suspended in it. And we're about to go on a tour of the cell. So we will talk about some of those subcellular structures, including things like lysosomes, which are a type of vesicle, mitochondria, and endoplasmic reticulum. These are the structures that we'll cover in our tour of the animal cell. It's not a comprehensive list, but it is a list of structures that are most important for us here in physiology. So all animal cells have membranes, both on the exterior of the cell and then membranes that occur inside the cell. They're just referred to generically as cell membranes. The membrane that surrounds the exterior of the cell specifically is called the plasma membrane. And then we have other cell membranes that surround the nucleus and that surround the exterior of organelles like vesicles, mitochondria, etc. I like this image because it does a good job of teaching the distinction between these two terms, cytoplasm and cytosol. Students can get hung up here, and let's prevent that. So cytoplasm on the left, cytoplasm is defined as the contents of a cell between the plasma membrane and the nuclear membrane. So notice that region of that cell is all highlighted in tan. Right? The cytoplasm includes cytosol, organelles, and other subcellular structures that are in the cell but outside the nucleus. That's cytoplasm. So it's this rich mixture of stuff. And we'll talk about what that stuff is in just a little bit. And then moving over to the right, well, what's cytosol? Notice that cytosol is the fluid that surrounds the subcellular structures outside of the nucleus. So different components of the cell are highlighted in tan there. Notice that the organelles are not part of cytosol, but they are part of cytoplasm. So cytosol is a gel-like fluid that occurs inside of our cells between the plasma membrane and the nuclear membrane. Our cells have cytoskeletons. I love this drawing. Because you can get a sense of the cell as being a three-dimensional thing. And the cytoskeleton is composed of three different types of protein filament. A filament is like a rod, three different types of protein rods that crisscross in all directions. And the cytoskeleton of the cell has many functions. Perhaps most obviously here, it gives physical structure, three-dimensional structure. It creates the shape of the cell, just like your endoskeleton creates the shape of you. These are the three different proteins that come in a rod shape that make up different cytoskeletal elements. And those cytoskeletal elements or filaments are actin filaments, intermediate filaments, and microtubules. And we won't really, in this course, we won't really distinguish among those three. They're just cytoskeletal elements. I'm happy with that. That's fine. I do want you to be aware, somewhere in your brain, tuck that away because you might need it down the road, perhaps in your microbiology class. We have these amazing stains that allow us to see very specific subcellular structures. And so what you're looking at here is a photomicrograph. It's a photograph taken through a microscope of two cells, and they have been stained using several different immunofluorescent stains. So one of these stains, it will bind to a very particular molecule within the cell. And then when we shine light on those cells that have a particular frequency of wavelengths, the stain gives off light or fluoresces. And so notice that we can see structures that are blue, structures that are fluorescing green, and structures that are fluorescing red, because three different immunofluorescent stains were used to colorize these cells. And the actin filaments, which are part of the cytoskeleton, are shown in green, and the microtubules, another part of the cytoskeleton, are shown in red. And then a third stain was used to highlight the nucleus of these cells in blue. And shown here on this slide are some of the functions of the cytoskeleton. So again, they give the cell a particular shape. The cytoskeleton is dynamic. These tubes can get, or these these rods can get longer, they can get shorter. So some of our body cells are able to move. Like our white blood cells can actually squeeze their way through these tiny openings in the walls of our tiny blood vessels called capillaries. And our white blood cells can maneuver through our tissues to a site of infection. The cytoskeleton is also responsible for moving materials within the cell. So some of these rods of protein, some of these cytoskeletal elements act as like a monorail, for example, and molecules will travel like you're traveling on BART or you're traveling on a train from one point in the cell to another point. And then these cytoskeletal elements also connect to the plasma membrane, and they play an important role in giving structural stability to a tissue. So let's say we have a group of skin cells, for example, in your epidermis. In the deeper layers of your epidermis, those cells are connected to each other. Their plasma membranes are connected to each other, and internally, inside those cells, the plasma membrane is connected to cytoskeletal elements. So those multiple cells in a row can act as a structurally functional body of cells because they're connected to each other and they are connected to their cytoskeletons. The nucleus. The nucleus is sometimes referred to as the brain of the cell. I disagree with that. I really think the plasma membrane calls the shots, determines what that cell is going to be doing from one moment to the next. But here we're looking at the nucleus, and the nucleus is surrounded by the nuclear envelope, sometimes called the nuclear membrane. It is a cell membrane, so it's made out of phospholipids with proteins embedded in it, and there are holes in that membrane that allow substances to enter and to leave the nucleus. And inside the nucleus is where we find the DNA that you inherited from both of your parents. And DNA takes a couple of different forms, and chromatin is the loose or the unraveled, the stringy form of DNA. And later on, we'll talk about chromosomes. Those are the tightly coiled little bundles of DNA. So in this nucleus, we see the uncoiled DNA, and we call that chromatin. And then there's a nucleolus, and the nucleolus is where certain types of RNA are produced. Outside the nucleus, as part of the cytoplasm, we find molecules called ribosomes. Ribosomes are made out of RNA, ribosomal RNA, and ribosomes play an important role in protein synthesis, which we'll talk about in just a little bit. And proteins can be synthesized using these ribosomes that are either free ribosomes, meaning they're floating around in the cytosol, or ribosomes that are part of rough endoplasmic reticulum. They are attached to a membranous organelle, and these ribosomes, whether they're free ribosomes or part of the rough endoplasmic reticulum, ribosomes play a role in protein synthesis, and we'll look at that very soon. This word organelle, I've used it several times already today, and an organelle is a subcellular compartment where specialized functions occur. And so all organelles are surrounded by cell membranes. Mitochondria are an example of an organelle. Mitochondria have to be my favorite organelle because they have this super cool backstory. Way before animal cells existed, before eukaryotic cells existed, for the first billion years of life on the planet, all cells were prokaryotes. So think bacteria and archaea. Prokaryotic cells are much smaller. They don't have any organelles. They don't even have a nucleus. So there's very there's no compartmentalization inside the cell. And so in the origin story of eukaryotic cells, mitochondria play an important role. They used to be free-living prokaryotic cells. Mitochondria used to be free-living bacteria-like cells, and they retain some of those characteristics today. Our mitochondria today still have their own DNA, and their membranes have a lot of biochemical characteristics in common with bacterial membranes rather than animal cell membranes. And so what do mitochondria do for us today in the modern eukaryotic cell? They are the site where most of our ATP is produced. So sometimes mitochondria are referred to as the powerhouse of the cell because they are super important for producing not all, but the vast majority of our ATP. Here's another photomicrograph. This was again taken using a transmission electron microscope, where we can see the nuclear membrane there. And in the cytosol, we see many mitochondria. So one of your cells has many mitochondria, and the exact number of mitochondria depends on the function of that cell. How big is the demand for ATP? So muscle cells, for example, have a huge demand for ATP. They are some of the most metabolically active cells in our body. So muscle cells will have thousands of mitochondria, and we can contrast that with a fat cell. Fat cells aren't particularly metabolically active. They need to maintain homeostasis, so they do have some mitochondria, but they don't need as much ATP, so they have far fewer mitochondria. Form follows function. The two organelles shown here are endoplasmic reticulum, or ER. The rough endoplasmic reticulum is studded with ribosomes, and when you look at it through the microscope, it has a rough appearance, hence the name. And the smooth endoplasmic reticulum does not have ribosomes, so it looks smooth. And what endoplasmic reticulum, along with the Golgi apparatus, and we'll talk about that next, what they all have in common is they have a membranous structure that's sort of like elaborately shaped, flattened sacs. Let's talk about what each of these does. The rough endoplasmic reticulum is responsible for helping to manufacture proteins, and then these proteins are packaged in a lipid coating and then sent off often to the Golgi apparatus for further processing. The smooth endoplasmic reticulum is where our cells manufacture lipids. Cholesterol is a lipid. Phospholipids are lipids. There are many other lipids that are used inside the cell as well as secreted for use elsewhere outside the cell. The Golgi apparatus has a similar structure, this elaborate flattened sac structure. The Golgi apparatus is akin to the cell's distribution center. So newly synthesized proteins, for example, will be shipped over from the rough endoplasmic reticulum to the Golgi apparatus, and the Golgi apparatus will essentially package it in a way that has a molecular address on it, like a mailing label. Where should this protein go? Should this protein go to the plasma membrane? Should this protein go to the nucleus, etc.? Here's another image of the Golgi apparatus, which is sometimes called the Golgi complex. And we can watch a newly formed protein. So up top, that number 1 and number 2 and number 3, that's a newly manufactured protein moving toward the Golgi complex. And as it moves through the Golgi complex, it gets repackaged and with a destination, a molecular destination attached to it. So if we see 789, that newly synthesized protein gets to wherever it needs to go, whatever function it's going to serve in the cell, it goes there. Vesicles. There are many different types of vesicles inside of our cells, and a vesicle is just a spherical sac. It's a storage container where the walls of the container are made out of cell membrane, a double layer of phospholipid. And vesicles are named according to their contents. What's inside that vesicle? And whatever is inside that vesicle determines the function of that vesicle. So I picked this image because we see several different types of vesicles here. Endocytic vesicles carry material that was recently brought into the cell by a process known as endocytosis. Lysosomes contain a variety of enzymes. At the bottom, we see peroxisomes. That's a different type of vesicle, and they contain a different collection of enzymes for different purposes. It can often be difficult to tell what type of vesicle is what until you know the contents of the vesicle. So we'll move on to the details of protein synthesis. As I mentioned earlier, proteins are everywhere in the cell, and many of them function as little molecular machines. Life as we know it couldn't exist without proteins. So when scientists engage in the search for signs of life outside of our planet, like on Mars, for example, they are very interested in finding amino acids and evidence of proteins. Life as we know it wouldn't exist without proteins. All of your cells, except your red blood cells, all of your cells are constantly engaged in manufacturing new proteins and breaking down old proteins. So inside all of your cells, there's this protein turnover that's happening 24/7. Old proteins can get degraded, and so to keep the proteins all in optimal working function, the older ones are broken down into amino acids, and then those amino acids can either leave the cell and be used elsewhere, or the cell can use those amino acids to synthesize new proteins. So we've seen this before. This flow of information is known as the central dogma of biology. And it just refers to the fact that DNA provides coded information. The sequence of bases in DNA, the A, T, G, C, provides a code for the synthesis, for the manufacture of particular molecules of RNA. And RNA is also a coded language. The sequence of bases in RNA provide instructions for manufacturing proteins. RNA, particularly messenger RNA, provides instructions for stringing together amino acids in a particular sequence. So I hope you remember that living things use 20 different amino acids, and messenger RNA provides the instructions for the order in which to string amino acids together in order to build a specific type of protein. So this is the central dogma. We now know that this doesn't always hold true. There are many regions of our genome. A lot of DNA doesn't code for anything, and sometimes it's called junk DNA, but we're still working to understand the purpose of these non-coding regions of our genome. And now we understand that there are many different types of RNA, and only one particular type of RNA, messenger RNA, provides instruction for making proteins. There are other types of RNA that are not involved in protein synthesis. They might be involved in regulating protein synthesis, but they don't code for proteins. So our understandings of the role of DNA and RNA have expanded dramatically over time, but the central dogma is still important. Though we now know that most of your genome, most of your nuclear DNA does not code for protein, and we now know that there are many different types of RNA, and RNA plays several different roles inside of our cells. So this is a basic overview of the process known as protein anabolism, building proteins. Proteins are large molecules made out of smaller building blocks, and the monomer subunit of proteins is the amino acid. So the two stages of protein synthesis are known as transcription and translation. And this pretty simple diagram shows you where each of those processes occurs. Transcription is step one, and it occurs in the nucleus. So let's look at the nucleus of this cell, and we see a molecule of DNA, and that little arrow that's labeled transcription indicates that that molecule of DNA is used to create a new molecule of RNA, and this is specifically messenger RNA or mRNA. That's a process of transcription, using a gene that's made out of DNA to create a new molecule of messenger RNA. That's transcription. What happens next is that molecule of RNA leaves the nucleus and enters the cytosol. In the cytosol is where step two occurs. Translation occurs in the cytosol, and it involves a molecule of RNA and a ribosome. That ribosome reads the RNA code, and a new protein is built. One amino acid at a time is added to a growing polypeptide chain, this long chain of many amino acids tacked together in sequence. And then that protein is going to fold. So to sum up this slide, the two processes that are involved in protein synthesis are step 1, transcription, and that occurs in the nucleus. Step 2, translation, and that occurs in the cytosol. Here's that flow of information. During transcription, we take a section of DNA called a gene, and that language of DNA is transcribed into the language of messenger RNA. Notice that DNA is a double-stranded molecule and RNA is a single-stranded molecule. Then in the cytosol of the cell, that RNA is translated from the language of a nucleic acid into the language of a protein. A gene is a section of DNA that codes for a particular sequence of RNA. So shown here, if we start on the left side of the image, we can see that chromosome coming from the cell nucleus. So this is a chunk of DNA that you inherited from one of your biological parents. And that chromosome is made up of DNA and proteins, tightly coiled DNA. It's coiled around proteins called histones. And if we uncoil a section of that DNA, and if we use that section to make a molecule of messenger RNA, that section of DNA is called a gene. That gene codes for a particular molecule of messenger RNA. This image shows you the process by which a gene is used as a template for the formation of a new molecule of messenger RNA. Nucleic acids rely on what's known as complementary base pairing. Perhaps you've heard about that in another biology class. For example, in a molecule of DNA, A always partners with T and C always partners with G. Well, we can use complementary base pairing to ensure that the right molecule of RNA is formed. C will always code for the placement of a G, and A will always code for the placement of a U in the new molecule of messenger RNA. I'm not going to spend much time on this slide because there's a mistake on it that I just caught, and perhaps you're familiar with the process of transcription and specifically translation. There is an error right here, so I'm just going to move on to the next slide. And if you're familiar with translation, maybe you can spot the error. We'll use the next slide instead. All right, so this image also shows the process of transcription and translation. And we see in the cytosol, we see a molecule of messenger RNA to which a ribosome is latched on. And the ribosome will slide along the messenger RNA and attach one amino acid at a time. So here we have our growing amino acid chain, and this process is known as translation. This is a different artistic representation of translation, where we have our messenger RNA molecule shown in pink, and our ribosome is shown in light blue, and our growing polypeptide chain is shown right here in sort of these gray, green, different amino acids. Every amino acid can be represented by a 3-letter abbreviation. So this is alanine and valine and leucine and glycine, etc. You do not need to know those abbreviations. And then we can get what's known as a polysome. And that means that a single molecule of messenger RNA, and what color is the messenger RNA in this diagram? Can you find it? Yeah, it's green. Good. Notice that that messenger RNA has several different ribosomes attached to it, and all of those ribosomes are simultaneously sliding along and reading the RNA code and building new proteins. So this ribosome up here on the left, this one is in the earliest stages. It just started to read the RNA, and there are no amino acids sticking out of it. This one's a little further along, and notice the polypeptide chain, the growing polypeptide chain is represented in purple. And this one is the furthest along, and When that ribosome reaches the end of the RNA molecule, the completed polypeptide chain will finish folding and protein synthesis is completed. This is a different artistic representation of translation. Notice that there is the blue messenger RNA molecule, and there are several ribosomes, or maybe that's meant to represent one ribosome as it slides along the messenger RNA. And we can see the polypeptide chain elongate as that ribosome makes its way from right to left. And so we can see the process of protein folding. And I'll use this slide to remind you that every protein has an amino, a terminal amino group. Sometimes it's called the N-terminus. And then every protein has a terminal carboxyl group, and sometimes it's called the C-terminus. So we can have a way of discussing one end of a protein versus another. In class, it's rare for me to show a video during a lecture meeting because our class time is precious and students can watch videos outside of class. But occasionally there is a video that is of high enough quality, meaning it clearly communicates important or complicated bits of information in a short period of time, and I judge that it's worthwhile using class time to watch the video. This is one of those videos. So I'm not sure how this will work for you, the viewer. Perhaps you can just click on the play button and this video will play for you. It's about 5 minutes long. If it doesn't play for you in this lecture recording, there's a link at the top, and I'll encourage you to spend the 5 minutes watching this excellent video that shows the process of protein synthesis in real time in our cells. So you can see how these molecules are interacting, and you can see how long it takes to go from gene to protein. The exact sequence of amino acids in a polypeptide chain will determine the overall three-dimensional shape of that folded protein. And form follows function. The three-dimensional shape of a protein is crucial for the ability of that protein to function properly. Here we see some of the forces, intramolecular forces and intermolecular forces, that help the polypeptide chain fold in a highly specific fashion. So intramolecular forces mean the forces that occur between atoms within the same molecule. So a polypeptide chain is one molecule. It's created when amino acids are linked together by peptide bonds, and different amino acids have different proclivities. They behave differently with their neighbors. Some amino acids attract each other and form bonds, and other amino acids repel each other. And then intermolecular forces refers to forces between atoms that exist in different molecules. So this polypeptide chain is surrounded by water molecules. They're not really represented here on the diagram. I can't point to a water molecule, but you know that water is polar. It plays the magnet game. And one of the ways in which one amino acid differs from another is in the polarity of that variable side chain. How well does it play the magnet game, the water game? That's another good name for it, but the magnet game. So some amino acids have many polar covalent bonds, and other amino acids have many non-polar covalent bonds, and so they tend to be hydrophobic. The primary structure of a protein refers to the exact sequence of amino acids. It's something that could be read off in a list. So let's say you're working in a coffee shop back in the good old days, pre-pandemic. You're doing your homework in a coffee shop, and somebody who's standing on line notices that you are studying proteins. And she says to you, "Oh, I'm a protein biologist. What are you studying?" And you tell her that you're studying for your physiology class that you absolutely love, and you ask her, "Oh, well, what do you study? What about proteins do you study?" And she says, "I'm working to understand how the primary structure of a protein determines how it folds." She means She's working to understand how the exact order of amino acids gives rise to a protein that has a particular three-dimensional shape. So she's interested in this order shown here. Now we're going from the beginning of this protein. I'll read from right to left. That's methionine and glycine and arginine, another arginine and isoleucine, on down. And then she is curious about, well, what happens during the black arrow? We know what the primary structure is. How do we get to this three-dimensional shape? One way that we can alter the primary structure of a polypeptide chain is through genetic mutation. As we've just been talking about, genes provide instructions for making proteins. If there's a change to the genetic code, if one or more of those base pairs in DNA is altered, it can give rise to a protein that has a different primary sequence, a different order of amino acids, and then that protein will fold differently, and it will have a different function. Depending on the extent of the chain, that protein may still be able to retain some of its function, or it might be completely non-functional. So if this mutated gene undergoes transcription followed by translation, it could lead to a mistake in the amino acid sequence, because in this DNA we see that two bases have been altered. A G has been changed to an A, and a T has been changed to a G. There are all sorts of genetic disorders that are caused by DNA that leads to the formation of a misshapen protein. And sickle cell disease is the classic example that we use for teaching purposes, because we understand the genetic basis for sickle cell disease. We understand that really well. We know what the changes in the DNA sequence are, and we know how those changes in the DNA sequence lead to a change in the primary structure of the polypeptide chain that gives rise to hemoglobin. So sickle cell disease is a genetic disorder that leads to a misshapen form of hemoglobin, and that misshapen form of hemoglobin can cause red blood cells to have a sickle shape, as seen in that lower image on the right. And so going through the DNA and the amino acid sequences on the left, we can see in typical hemoglobin at the top, we see for that region of the gene that codes for hemoglobin, in typical hemoglobin, we have a CTC which base pairs GAG. And that genetic code in that specific 3 base position provides instructions for the ribosome to insert a particular amino acid called glutamic acid during translation. If we look at the sickle cell gene, in that same position, we have our 3 bases CAC, which complementary base pairs with GTG, and that change gives rise to a different amino acid during translation. Instead of glutamic acid, a valine is put into that position. And that change from glutamic acid to valine in that position gives rise to the misshapen form of hemoglobin that's often referred to as sickle hemoglobin. And I showed you this image in a previous lecture. Up top we have typical hemoglobin, and down below we have sickle hemoglobin. Sickle hemoglobin has different physical properties. It's less water soluble, and so it gives rise to those sickle cells. Those sickle-shaped red blood cells don't travel particularly easily in capillaries. They get caught up around corners, they get caught up on each other, and they don't deliver oxygen as effectively as typical red blood cells do. Protein synthesis is happening in all of our cells all of the time, except red blood cells. Red blood cells don't have any nuclei. They eject their nucleus when they are in the process of being formed. So red blood cells don't have any nuclei, and without a nucleus, a cell doesn't have any DNA, so transcription can't happen. So red blood cells don't do protein synthesis, but the rest of your cells do, and they're doing it pretty much all the time. And there are perhaps 10,000 different proteins in your body and thousands of different proteins in an individual cell. And when those cells are forming a particular protein, and when those cells are degrading or catabolizing a particular protein, those processes are highly regulated. So in any particular cell in your body, just a handful of genes are turned on, meaning transcription is happening. And then translation follows transcription. And so transcription, protein synthesis can be controlled by controlling transcription. We can turn on transcription, we can turn off transcription for specific genes using specific sets of molecules. And these sets of molecules that turn on or turn off transcription, whether or not the cell is going to make that protein, those are called transcription factors. They're little switches. A transcription factor can initiate or turn off transcription. We can use this diagram to examine one type of transcription factor. And this transcription factor is created inside the cell when the cell receives a message from its external environment. So this is leading us toward mechanisms of cell signaling. How do cells send and receive and then respond to messages from other cells? This cell happens to be responding to a message that it received from some other cell. And it is one example of a whole class of transcription factors. So let's look at the diagram that's on the right. This will set us up for part of the cell signaling chapter. So here we see the plasma membrane of a cell. And embedded in that plasma membrane, this purple molecule here is a protein, and it's called a receptor protein. And like all proteins, it has a particular three-dimensional shape. This receptor protein's three-dimensional shape allows it to bind to a signaling molecule shown as sort of this red upside-down teardrop. This is a signaling molecule that was secreted by some other cell somewhere else in the body. And it floated around, probably in the bloodstream, and it landed on the receptor that's embedded in the exterior surface of this target cell, this recipient cell. And so when the signaling molecule binds to its receptor, it initiates this whole cascade of biochemical events that happen in the cell, where one thing leads to the formation of another, which causes the formation of a third thing, etc. It's called a cascade, and we'll talk more about it later. The outcome of this signaling pathway is the formation of a transcription factor. And that, in this case, that occurs inside the nucleus of the cell. And so this transcription factor can now bind to the DNA. It binds to a very specific region of DNA at the beginning of a gene that codes for a particular protein, and it will activate, look at this, it will activate the formation of messenger RNA. So this transcription factor happens to turn on transcription, and this messenger RNA will then leave the nucleus, translation will occur, and we'll have new protein. So ultimately, the cell signal way up here leads to or causes the formation of the protein that we don't see here, but if we could follow it through, we would see translation happening in the cytosol and a new protein would form. So this brings us to the end of the first of 4 segments for cells and movement across the plasma membrane. We just finished talking about the first 2 topics here, cell structure and function, and the very basics of protein synthesis. When we meet again, I'll describe to you in detail the structure of the plasma membrane and how particular materials get into and out of our cells. Thanks for watching.